The test cross was first introduced by Gregor Mendel, the father of modern genetics. It began with his study of the genetics of traits in pea plants. In theory, each organism carries 2 copies of each trait, one dominant and one recessive. The dominant trait determines the outward appearance of an organism, or the phenotype. The question is, which organisms with the dominant phenotype have 2 dominant alleles, and which have 1 dominant allele and 1 recessive allele? To answer this, we use the test cross. A test cross is a way to explore the genotype of an organism. It is an experimental cross involving mating an unknown genotypic individual with a known homozygous recessive. Hence, the phenotype of any offspring will reflect the genotype of the unknown parent. Test crosses can be used to determine whether a dominant phenotype is homozygous or heterozygous. If the unknown parent is homozygous dominant, all offspring will express the dominant phenotype. In contrast, if the unknown parent is heterozygous, half the offspring should be dominant and half recessive. A test cross between a heterozygous dominant parent and a recessive parent will produce offspring of a 1-to-1 ratio. A test cross between a homozygous dominant parent and a recessive parent gives all dominant progeny. And as example here, if we take a black cat and we want to determine the genotype of that cat, and we breed them with a white cat, here are the Punnett squares to determine whether that black cat is homozygous or heterozygous. And in our example here, the offspring, half of the offspring were black and the other half were white. So we know that the genotype of the unknown individual according to the offspring. So in this example, since half of the kittens are white and half of the kittens are black, that means that the cat is a heterozygous dominant.